Hello, and welcome to the New Jersey BioFoundry Showcase, hosted by the New Jersey Innovation Institute. My name is Will Lutz, and I'm the General Manager for Entrepreneurship and Commercialization at NJII. On behalf of everyone on our team, I want to thank you for joining us and checking out our cohort companies while they showcase their startups in cutting-edge biotechnology. Now, the New Jersey BioFoundry program is funded by a federal EDA I-6 grant. We want to thank our friends at the EDA for their continued support in making this program a reality. In addition to the EDA, a number of organizations and people have helped put this together as advisors, sponsors, coaches, and mentors. Now, there's just no way I can capture everyone up on the screen. There's just too many, but suffice to say, thank you so much for all of your help and support. We owe you a ton. We could never have gotten this far without you. Now, some of the organizations that have supported us are doing some really innovative stuff, and many of them are right here at the expo. I strongly recommend checking them out. A bit about BioFoundry. The program here is designed to support the best super early stage regenerative medicine companies that we can find. Over the past few months, we've been working with these killer companies on business growth and leadership. Our program provides support and mentorship around communications, fundraising, marketing, hiring, and network building, all of which are critical elements to a successful biopharmaceutical startup. Each of these companies graduates from our program with our endorsement for further success. All right, that's really enough for me. Let's get into it. Let the showcase begin.
Hello, my name is Ryan Myers, and I'm the CEO and co-founder of AdGraft Therapeutics, where we're deploying cutting-edge genetic treatments in order to change lives. And few conditions affect more lives in the world and in the USA than addiction. Addiction costs the USA over $1 trillion every year. That's roughly 6% of the US GDP and 17% of all US government spending. And unfortunately, one of the largest issues within addiction or substance use disorder is alcohol use disorder. In fact, there are 16.3 million Americans diagnosed with alcohol use disorder, of which the US spends roughly $224 billion a year to treat. And unfortunately, it results in over 88,000 deaths a year. Now, on top of all this, the problem is growing of AUD. In fact, alcohol consumption since 1998 is up roughly 18%. And this all might seem terrible, but the worst part of all this is that of every 4 patients that go to seek some kind of treatment for alcohol use disorder, roughly 3 of them will relapse within the first year. That's where AdGraft hopes to step in. AdGraft's unique treatment technology turns the patient's skin into a bioengine. And this bioengine is capable of producing therapy and deploying therapy directly into the bloodstream. In the case of AUD, we're able to deploy GLP-1, and this is how we do it. We start off by taking a patient, and we take roughly a fingernail-sized sample of their skin using a procedure similar to a mole removal. We then move that sample of skin to a laboratory where we genetically modify the skin using common Cas9 CRISPR on the stem cell level. We then separate out the successfully edited skin cells and culture them for roughly 4 weeks. In this 4-week period, they grow roughly to the size of 2 credit cards. We then take this 2 credit card size skin graft and transport it to a hospital location, where we will then re-implant that skin graft through a normal skin graft procedure back into the original donor. This skin now can produce and deploy medication directly into your bloodstream and give positive therapeutic effects as a result. The GLP-1 utilized is able to help you to regulate your dopamine levels and shows extremely positive effects when we talk about the major pillars of treating AUD. The 3 major pillars of treating AUD are preventing addictive behavior formation, preventing relapse, and reducing active drinking. In the case of preventing addictive behavior formation, in our preclinical mouse models, we proved that the treated mouse population showed significantly lower levels of addictive behavior formation, the blue being the treated and the gray being untreated. In the case of relapse, and this is really the gold standard, we exposed mice on day 1 to an environment where they could choose alcohol use or not. Day 7, we removed them, implement the procedure, and on day 26, we reintroduced them back into the environment. Day 27, we measure response. As you can see, relapse was significantly decreased in the individuals that were given the treatment, and this is what you're looking for in a relapse model. In the case of reducing active drinking, this is what we call the parachute effect. And the goal is to, in the case of relapse, prevent harmful relapse events, which can often be the most dangerous to patients. And we demonstrated the ability to reduce active drinking. In fact, if my partner, Dr. Ming Xu, could share this presentation with me, he'd tell you that in 25 years of studying substance use disorder, these are some of the most promising results he's ever seen. And he's not the only one excited about this. In fact, some of the major scientific publications around the world have published and corroborated our results on AUD, cocaine use disorder, nicotine use disorder, type 2 diabetes, and acute obesity. And the reason this platform is so exciting and works so well is because we're able to stably supply therapeutic molecules that weren't previously very viable or useful because maybe they have a short half-life or they can't accumulate in the body to a high enough degree. Our ability to use the bioengine stably supplies the GLP-1 molecule to the bloodstream and gives therapeutic effects. Additionally, the autograft minimizes immune response by your own skin being reimplanted, lack of rejection possibility. The procedure is long-lasting, highly effective, and minimally invasive, and the graft should act as a normal skin graft does for, say, a burn victim. And finally, our extremely encouraging preclinical results give us high hopes that it's translating well into humans. Beyond the technology itself, in the form of abuse, there's extreme benefits to the flexibility of the platform. This single treatment for alcohol use disorder also effectively treats nicotine use disorder, cocaine use disorder, and co-abuse between the three. This is extremely promising considering that multiple patients going through alcohol use disorder might also have a secondary disorder that makes treating either one more difficult. Our platform coverages secondary abuse development and gives extremely positive results for treating abuse, as we showed earlier. And this is really encouraging because the current AUD landscape, quite frankly, is not great for patients. Some treatments quite don't work when tested against placebo, and some do work but have highly toxic side effects. AdGraft has the possibility to be an extremely effective but very low toxicity one-time treatment that really will be able to help patients. When it comes to the market, we're anticipating that a total population of 16.3 million with the disorder. Currently, unfortunately, only 9% are using treatment. That's because the treatment options aren't great. We hope to expand this number, but if we're conservatively estimating a market, we anticipate being able to capture between 5 and 20% of the market currently utilizing treatment, which is standard for a new procedure. And that leads us to a $5 to $29 billion market in AUD alone. As you can see, nicotine and cocaine also provide positive potential future markets as well after our lead indication of AUD. Our management team is highly equipped and capable of addressing this problem with close ties to the University of Chicago. I myself have experience as the CEO in BC and healthcare management consulting, and also in completing my MBA from the University of Chicago Booth. Dr. Ming Xu, our chief science officer, is a professor at the University of Chicago and an expert in neurobiology and substance use disorder. He has over 100 publications, and alongside Dr. Xiaoyang Wu, co-invented this platform. Dr. Wu is an associate professor at the University of Chicago Pritzker School of Medicine, an expert in tissue engineering and regenerative medicine, and once again invented the platform. Our advisory board is well equipped with experts in the substance use disorder realm. Michael Darcy, former 30-year CEO of the Gateway Foundation, and Dimitra is an experienced C-suite biotech executive. We raised our pre-seed through the help of Limitless Ventures and Chicago Booth Angel Network. And we have other affiliations as well. So we've worked with the Creative Destruction Lab, currently in the BioFoundry, and completed Yale Innovation to Impact program. We were named 2021 Global Deep Tech Pioneers and have an unofficial partnership at this time with the Gateway Foundation as we go forward in developing our treatment. As of today, we're working to engage the FDA. And in fact, we're hoping to engage the FDA in Q2 of 2022 and begin raising a seed round shortly after that in order to fund our preclinical data package. And that data package will go a long way for not just taking forward the AUD platform, but all of our substance use disorder platforms. We're currently still in the discovery phase for opioids, but we're proving concept in nicotine, and we published results for alcohol, cocaine, and actually alcohol, cocaine co-use. Currently, we are engaging the FDA, and that's our major focus. But outside, we're also proved concept in type 2 diabetes and acute obesity. We're working on discovery for cognitive decline and a number of other disorders as well. So please, if you'd like to talk about our progress, if you'd like to talk about clinical partnerships, we would love to continue the conversation. My contact information is right here, and please come see us at our booth as well. Thank you very much.
Thank you for the opportunity to present today. The question I ask you is, do you know anyone who's had cancer? What it feels like to be diagnosed with cancer? I want to tell you about a new therapeutic category called cell therapeutics. They're seeking and achieving cures for cancer, but at very high cost with significant manufacturing problems. Raven Biomaterials can impact these manufacturing problems with our transformative separation solution. We solve critical cell therapy manufacturing and patient monitoring challenges, increasing yield, decreasing production time, and improving the quality of therapeutic and diagnostic testing. All of this improves the possibility of more patients to have access to life-saving therapeutics by significantly decreasing the cost, and provides an investor with a therapeutic-grade investment with multiple returns within 5 years without the regulatory risk. Magnetic bead separation is used today for many processes, but we initially target cell therapeutics because it has a problem we can solve, but also because it's an explosive market, attracting over $1.1 billion in investment since before 2018, racing to approve products and expanding at an exponential rate. Our initial customers will be the 700 companies approaching or in early stage trials. These aren't your common everyday startups. Many have raised over $50 million. Just focusing on a sliver of today's market opportunity, the market for RBI beads is an estimate of $1.4 billion split between manufacturing and patient monitoring. We focus on replacing an existing required product arena with a less expensive, higher performing alternative. Cell therapy manufacturing relies on magnetic cell separation, and yet every time they're used, there is significant cell loss in harvest, transport, selection, modification, expansion, and in final purification. Current manufacturing inefficiencies are well recognized and provide Raven with a great opportunity. The therapeutic goal is to modify and manufacture an adequate number of cells to provide a therapeutic dose. And yet cell loss adds unnecessary expense that drives up the price of therapeutic treatment and increases time to treatment. Adding weeks to receive a therapeutic can be a death sentence to a late-stage cancer patient. Raven's RBI bead innovation simply outperforms the competition, providing over 90% cellular recovery in a fraction of the processing time without any pre-processing steps. This is for a single separation process. During manufacturing, separation processes are needed multiple times, each time multiplying the cell and hence therapeutic loss. After 3 separations, we're still in the 90s, while our competition is much lower and much slower. All of this is due to the patented power of the RBI core. Raven's RBI beads are made of a solid core of metallic microparticles that are 50 times more magnetic, 4 times more dense, which we coat with a thin film to directly bind to antibodies. Others have a polymer construct in which they encapsulate numerous, much smaller iron oxide particles that are weakly magnetic, making them less effective and requiring longer binding change to bind their antibodies. These proprietary material properties give us distinct technical benefits, with resulting manufacturing benefits. Our core material properties enable Raven to uniquely process in larger volumes of more complex biological mixtures, provide a higher yield of high-quality products and diagnostic resolution, with a significant financial benefit, not only directly in yield, but also quality, processing efficiencies, and overall cost of processing equipment. RBI beads provide a cleaner, cell-based material. They can be used both for depletion, that is removal of unwanted cells, or positive selection. But our first focus is depletion, because it doesn't alter the desired cells and isn't left in the therapeutic material. Our product does not require FDA approval, as it will be used as a GMP ancillary material for manufacturing and a supply material for patient monitoring. Raven targets the $1.2 billion market improving cell therapeutics manufacturing. Recognizing the proprietary nature of cell therapy manufacturing, we structure our product launches to build relationships. First, providing a cleaner starting material with more cells. Second, to purify the therapeutic at the end of the process without loss. Customers have indicated this is a great but more proprietary need, but an area in which we can deliver. Third, there are great opportunities to improve processing by removing residuals, untransfected cells, viral vectors, unwanted proteins that remain in the system after each step. With Raven's RBI beads, we significantly increase yield, provide cleaner, high-quality product production, and the best part is we do this much faster than the competition can in larger volumes. The same RBI beads will be used to improve patient monitoring, getting us closer to larger customers faster. All of the approved products today are required by the FDA to monitor treated patients for 10 to 15 years for durability of therapeutic and a continued cancer-free state. As we prove our capability, the doors open to us to work with approved products for their patient monitoring. While manufacturing is our target market, patient monitoring is a $230 million market that can provide earlier beneficial cash flow. With funding and ability to staff and scale appropriately, Raven's opportunity grows as the therapeutic category grows. Founded at the start of 2021, we obtained seed funding, completed proof of concept development, and have 2 products ready for sample testing by customers. In the next 2 years, we'll focus on customer trials and collaborations, and expanding our product and patent portfolio, such that we're positioned by 2024 to support our first customer in a clinical trial and launch our patient monitoring products. We believe that in less than 5 years, we can be poised to garner a significant market share. With a recognized superior product, demonstrated customer traction, we can reach profitability and be poised for exit or IPO. Our focused cell therapy plan builds our core competencies in manufacturing, handling cells, and diagnostics, which open up doors in other markets. Our team is ready to execute the plan. I've spent 30 years in the medical space working repeatedly to use novel innovations to drive commercial success. I've surrounded myself with a team with great track records in all the right areas: immunology, magnetic beads, blood, and therapeutic processing, from both the scientific and the commercialization aspect. With adequate funding, we can take this opportunity, staff, and scale appropriately to meet our goals. Raven Biomaterials is poised as a transformative separation solution, initially solving critical cell therapy challenges. If you're interested in learning more, please reach out to me directly. Thank you for your time.
Hello, thank you for giving me this opportunity to explain what Gilead is doing to personalize therapies for autoimmunity. Autoimmunity is a growing problem. It's an invisible epidemic with about 30 million Americans afflicted with various autoimmune diseases and not many treatment options available. Current therapies have major side effects. Steroids can be used, but only for the short term. Immunosuppressive drugs work, but they have strong side effects, including things like serious allergic reactions, increased risk for certain cancers. Even death is possible when you use these drugs. Most autoimmune therapeutics target the T cell function, but not the T cell receptor. Gilead would be the first to focus on the TCR. You have millions of T cells in your blood, and each one of them has a receptor that can bind to specific antigens. When the T cell recognizes a viral antigen, it will multiply and also kill the cells that are presenting it. This is how your body is protected by the T cells from infections. Gilead can now sequence the TCR accurately, and this is based on the technology we call T-seq that has a patent. So our technology consists of algorithms to analyze the data from T-seq, a healthy database of reference data sets that we have collected over time. And together with T-seq, these form the immunomap. And in combination with data measured in patients, we can personalize therapies, we can do dosage optimization, we can monitor the progress of diseases. The T-seq works on many different animals. It can be used in dogs, cats, pigs, rats, mice, monkeys, and humans. This is a big advantage because currently dog and cat models are being considered for studying human therapies, but also we can use our platform in the pet industry to treat pets and develop new treatments. So there's a virtuous circle here. Human therapies can be used to help pets, and pet therapies can in turn help humans. And dog and cat models can be used to develop new therapies, both for pets and humans. The market is immense. The human market is about 100 billion, and many, the biggest drugs are targeting autoimmunity, and it's a growing market. The pet industry is relatively new. There is only one drug right now that targets autoimmunity using immunosuppression, and that's for canine atopic dermatitis. The drug is called Cytopoint from Zoetis, and their market is about $1 billion right now. And it has been very successful in treating dogs for this condition. So currently we have one customer for our dog-cat offerings. We are helping develop new autoimmune diagnostics based on the T-cell receptor repertoire, and we want to develop new autoimmune therapies for them. Our goal in the next 2 years is to develop our healthy dog database and a diseased dog database and algorithms to analyze the data that comes from our dog work. We want to optimize existing therapies for dogs and also develop new therapies based on our technology. We hope to get revenue from vets by using our platform to help treat dogs and improve the treatments and personalize the treatments for each dog. We are two co-founders, Anita and myself. Anita is a PhD in biomedical genetics. She has over 10 years of experience in leading teams in academia and industry, and has built several assays in deep sequencing and has 5 patents that are form the basis of Gilead's IP. I have a PhD in physics. I've worked in the first map of human SNPs and have extensive experience in genomics, bioinformatics, and technology development. I've also co-authored over 120 papers and have 3 patents that are part of Gilead's portfolio. Thank you.
I'm Steve Navran, CEO and co-founder of Animatus Biosciences. When we founded the company a few years ago, we were intending to use reprogrammed stem cells to treat heart failure. We did have some success with this, but eventually realized that this approach was not optimal for commercialization. So we decided to develop a treatment that would help the heart heal itself. Before I talk about that, I'd like to give you some background on heart failure. Heart failure is a leading cause of morbidity and mortality in the developed world. There's an enormous amount of money spent on these patients, and it is an increasing problem with an expanding number of new cases each year. Consequently, a cure for heart failure would have a huge revenue potential. The reason heart failure is such a problem is that adult cardiac muscle cells don't replicate. This means after a heart attack, the heart can't produce new cells to replace the dead cells. Currently used drugs can only reduce the workload on the heart or make it beat more forcefully, but none of these drugs can repair the damage. Our new approach is based on cardiac developmental biology. In the embryo and early neonate, heart cells are able to replicate in order to construct the heart. Shortly after birth, these cells switch to a non-replicated contractile form. Animatus has developed 2 synthetic messenger RNAs that can reprogram adult cardiomyocytes back to a more primitive and replicated form, which can then regenerate a failing heart. We tested our messenger RNA combination in a mouse infarct model. This figure shows cross sections of mouse left ventricles that have been infarcted. The mouse that received the messenger RNA shows complete repair of the damage after 4 weeks. Interestingly, fibrosis was greatly reduced as well. When we looked at functional responses in these mice, we saw substantial improvement in pumping capacity 4 weeks after the infarct in the treated mice, while untreated mice continued to deteriorate. We think our messenger RNA combination has a number of advantages over our competition. For example, in heart failure, this is considered, in heart transplant, this is considered a cure for heart failure. Unfortunately, there aren't enough donor hearts to even touch this problem, so it's really not practical. Currently, adult stem cells and pluripotent stem cells are of interest. In terms of adult stem cells, there are some clinical trials going on right now, and they have shown some moderate improvements. However, they don't make new cardiomyocytes. They only moderate the inflammation after a heart attack. Because they have to be derived from donors and extensively cultured, they have high costs and are low in scalability. Pluripotent cells can be reprogrammed into cardiomyocytes, but they have the same cost and scalability problems of adult stem cells. By contrast, our messenger RNA combination has high efficacy in our animal infarct study, and we are very confident that this can be translated into humans. The cost of making messenger RNA is quite low compared to stem cells, and it also can be scaled up very well. And these two things have been shown in the messenger RNA vaccines that are currently being used against the coronavirus. We have a strong patent portfolio with several new patents in preparation. We're constantly making new findings that we think we can add to our intellectual property. Our developmental plan in the coming year is to raise a Series A investment and also complete a pig infarct study. The pig infarct study is important because the pig heart is very similar to the human heart, and this is considered a prelude to being able to do a human trial. We've also talked to a number of large pharmas about licensing our technology or partnering with them, and most of them have indicated that they would like to see a pig study before committing to us. We hope to start our clinical trials in 2024 and complete them by 2026. We realize that this looks like a pretty aggressive timeline. The FDA has an expedited review for high priority unmet needs, and we think that heart failure absolutely qualifies for that. We have a well-prepared management team with extensive experience in basic sciences and the biotech industry. We also have a distinguished group of physician scientists on our scientific advisory board who've been very helpful to us. If you would like to know more about Animatus Biosciences, please look at our website or email us, and I would be glad to answer any questions that you might have. Thank you.
Hello, everyone. My name is Joshua Yang, and I am the co-founder and CEO of Glyphic Biotechnologies, where we are developing a next-generation protein sequencer. Imagine if we could diagnose Alzheimer's decades in advance of symptoms, predict and prevent heart attacks before they occur, create personalized precision cancer vaccines for patients of all backgrounds, and engineer novel life-saving antibody therapies more easily. We are limited in our ability to do this today because proteomic analysis tools themselves are limited. Tools today are insensitive and not powerful enough to measure low abundance proteins. Tools today are low throughput in how many types and quantity of proteins that are measurable. Tools today also have complex workflows with little lab-to-lab consistency, requiring highly skilled operators to use them, which puts them out of reach for the vast majority of research laboratories. And finally, there are trade-offs. While some tools may provide adequate sensitivity, throughput, or ease, none provide all simultaneously. Glyphic is here to change that with our solution, a complete protein sequencing platform. This platform will be ultra-high throughput, measuring billions versus thousands of proteins. The platform will be at the single molecule sensitivity, enabling measurement of the lowest abundance proteins. And the platform will be a plug-and-play instrument. If you can pipette, you can sequence proteins. The way that this technology works is shown in the following. Here we see a glass slide with many different proteins from a sample adsorbed onto the glass slide. We will now focus on a single protein on this glass slide, but this reaction that I will describe is being done in parallel to the billions of proteins simultaneously. We can see here in this pink square one peptide that has been attached to the glass slide, and we have developed a novel chemistry, ClickP, that attaches to the last amino acid on the protein chain. This then causes a local tethering event to occur, where the protein is now connected at both the N and the C terminus to the glass slide. Finally, we cause this protein to undergo an N-terminal cleavage event, which liberates that last amino acid from the rest of the protein chain. You can see that the protein chain is one less amino acid long, but nearby, there is the amino acid connected to the ClickP molecule. Finally, we use one of our 20 high-affinity amino acid binders that can bind to each of the amino acids with high specificity and affinity, thus identifying it with a specific fluorophore. Finally, if you iterate throughout the cycle continuously for the entire length of the proteins on the glass slide, you will then have sequenced the entire set of the billions of proteins on that glass slide, and thus able to identify all of the billions of proteins in your sample. Next generation protein sequencing currently remains unavailable. There are companies that are like Nautilus and Dreamport that have proteomic solutions that they are developing, but these do not get you to the protein sequencing level data that you would need. Erysion is a competitor using a fluorosequencing approach. Their approach, however, is incomplete based on a fingerprinting mechanism that is not scalable to all 20 amino acids inherent to the technology. Quantum-Si and Encodia are two competitors that are developing protein sequencing technologies, but the feasibility of their technology has not been demonstrated. As such, no competitor has produced a commercially available product yet. We believe that Glyphic Biotechnologies will be able to access the large global proteomics market currently valued at $25 billion. However, we believe that this is only the immediate market opportunity, and we believe that revolutionary technology such as next-generation protein sequencing would enable a rapid expansion of the market through a long-term transformation of healthcare. We believe that applications in precision medicine, clinical diagnostics, and therapeutics development will enable the growth and market expansion opportunity that we plan to catalyze and penetrate. We have a proven approach to our business model. We are focusing on 4 customer segments: biotech companies, biopharmaceuticals, academic institutions, and CROs. And to these customers, we will offer them services, acting as an interim CRO, and an equipment, acting on the razor blade model with sales of integrated systems, consumables, instrument service plans, and training. We know this model will work because this is the same approach that research tools giants to date have taken, such as Illumina, who 5 years after launching their first sequencer product had achieved over $325 million in revenue and are now the genomics market leader. The same model taken by 10x Genomics, where after launching their first product, were able to achieve almost $250 million in revenue after 5 years and are now the single-cell market leader. No competitors have a working next-generation protein sequencer, and we believe that Glyphic Biotechnologies will be that proteomics market leader able to achieve similarly high revenue as these companies have with our proteomics solution becoming the proteomics market leader. And so I wanted to thank you for your time. We are Glyphic Biotechnologies, revolutionizing science, one amino acid at a time. If the story resonated with you, please feel free to reach out to me at joshua@glyphic.bio. We'd be more than happy to talk about ways we can work together to revolutionize proteomics and life sciences.
Woo! That was phenomenal. Kudos, good job to all of our BioFoundry teams. It has been such a pleasure working with you. And we are so glad that all of you could join us today and check out our pitches. Please stick around and chat with our founders and all of our partners that are out in the expo hall. Be well, be safe, be healthy.